All right, this is your chapter 12, Notes on the Cell Cycle. The role of cell division. For prokaryotes, this is how a new organism is made. For eukaryotes, growth, renewal, and repair for replacing cells. So cells divide in prokaryotic cells. This is going to be things like bacteria, because they are single cell organisms and they do not have any other way of reproduction. This is their method of reproduction. For eukaryotic cells, that is not the case. They cannot reproduce simply as and so that's going to be more for the purposes of growing, replacing cells, and repairing cells that are damaged. So this just shows reproduction. An amoeba would be an example here. Why is my pen not working? Would be an example there of something that would amoeba would be an example of a single cell organism that would reproduce by cell division. They are asexual organisms. Apparently my pen is not going to work. Let's see if I can figure out how to get that to work. Okay, we're just going to have to go without the pen this time, which is kind of annoying, but whatever. Okay, so this would be an example of amoeba in this first picture, dividing, and then the growth and development would be an example of like an embryo that's developing from that single cell, and then tissue renewal would be like replacing your stem cells very frequently. Not all of your cells will renew, but some of them will. Cell cycle is defined as the life of a cell from the time it is first formed from a dividing parent cell until its own division into two daughter cells. So think of it like parent and then the offspring of that. So this is not organism reproduction. We're talking about the reproduction of individual cells. The breakdown of the normal cell cycle plays a major role in cancer development and thus is an area of active research. And we're going to talk more about cancer here in a little bit. The results of cell division of eukaryotic cells. Mitosis produces two identical cells. So think about your skin cells. When they divide and make new ones, you want them to be exactly like the other skin cells. You don't want them to be any different. The other type of cell division we're going to talk about is meiosis. That's a special type of cell division that produces the egg and the sperm. And those will be used then in reproduction. So meiosis is only used in the male or female reproductive system. So in the male testes and in the female ovaries is the only time you'll see meiosis occurring. The rest of the cells will undergo mitosis. Organization of the genetic material. Prokaryotes, DNA is typically a single molecule and circular. In eukaryotes, the DNA is multiple pieces and is divided into structures called chromosomes. A typical human cell has over 2 meters of DNA, which is much longer than the cell's diameter. Thus, packaging into chromosomes will save space. So you won't actually see chromosomes until the cell begins division. Before that, the DNA is called chromatin, and it is going to be just loosely tangled inside the nucleus. Once the cell gets into that division process, there in prophase, it'll coil up or condense into these physical structures called chromosomes. So you can see chromosomes underneath a microscope. They're visible because they're so thick with all that DNA. But you can't see them until the cell goes into the division process. So when we typically look at cells in a microscope, you won't see them chromosomes. You'll just see the nucleus that's stained kind of dark because it has a bunch of DNA in it. And so this is a picture of a prophase cell where you've got the DNA that is condensed together to form the chromosomes. That's why that's so dark on the inside of the cell. Each chromosome consists of long linear DNA associated with proteins. Those are called histones. Chromatin is the DNA and proteins that build the chromosomes. DNA is not in chromosome format until the cell division begins. So chromatin is this loosely packed DNA around proteins. And then once that we enter into the cell division, into the prophase, those the DNA will come together in these chromosome structures. Each organism has a different number of chromosomes. Somatic cells are also known as body cells, and they're all the body cells except the reproductive cells. The reproductive cells are what we call gametes. That would be egg or sperm. In humans, somatic cells contain 46 chromosomes, 23 pairs. You get a full set of 23 from the egg and a full set of 23 from the sperm, and that is the initial, what we call zygote, and that zygote divides from one cell into 2, and 4, and 8, and 16, and 32, and on and on until you have trillions of cells that make you. In humans, gametes contain 23 chromosomes. That would be your egg or your sperm. All DNA must be copied, or we call that DNA replication, and we learn that process later, before a cell can divide. And if you think about it, that makes sense. One cell has 46 chromosomes. In order for each of the new cells to both have 46 chromosomes, you have to copy all of that DNA. After the DNA is copied, DNA condenses into chromosomes. This makes it so thick that it is visible under the light microscope. Each duplicated chromosome is called a sister chromatid and are joined together by a centromere. These sister chromatids will separate later in the division process so that each daughter cell gets a copy of the genetic material. Once the sister chromatids separate, they are called individual chromosomes. So here is a picture of a chromosome. Notice that they're attached together in the center by a centromere. The two sister chromosomes are each one half. Those two sister chromatids are identical to each other. So where do you get that? Remember, your DNA made a copy of itself during interphase. We haven't gotten to interphase yet, but DNA makes a copy of itself before the cell divides. And so one half is a copy of the other half. And those two will split apart during the division process. So here we start with a chromosome in step 1. DNA replicates. Now you have 2 of them, and now they are called sister chromatids. And then later they will separate, and now they're called individual chromosomes again. The separation of the chromosomes, or division of the nucleus, is followed by cytokinesis, the division of the cytoplasm. So let's talk about the cell cycle, the life of the cell. We did a graphic organizer in class over this. Mitotic phase, or the M phase, includes mitosis and cytokinesis. That's actually the division process of the cell cycle. Interphase accounts for about 90% of the cell cycle. Interphase is broken into G1, S, and G2. S is where DNA is copied. Most of the cell's life is spent in interphase. There are some cells that spend forever in interphase because they don't ever divide. For example, brain cells. You don't want to kill those with drugs because they won't come back. They are there and they rarely ever, if ever, divide. Muscle cells very rarely divide, but they can divide if they need to be replaced or they're damaged. So a lot of time spent in interphase. Skin cells divide very, very frequently, but they still spend a lot of time in interphase. So for example, if the life of a skin cell is 24 hours, 90% of that is going to be spent in interphase. So I think that's about 18 hours, and then the other 6 hours will be spent actually physically doing the division process. So this shows something similar to what we did in class. G0 is another stage that we talked about. Cells divide at different rates. Some cells don't divide at all or very rarely the entire life of an organism. These cells will either stay in the G1 and never progress and copy their DNA, or they will go into what's called a G0 phase in which they don't divide. Mitosis is divided into prophase, prometaphase, metaphase, anaphase, and telophase. You probably remember this from Biology 1, but you probably only remember PMAT, prophase, metaphase, anaphase, and telophase. Prometaphase is, we've kind of added that this year because it's kind of an important in between prophase and metaphase, because it's a big step to go from prophase to metaphase. And so prophetamase kind of links those two together. So in prophase, the chromosomes, I'm going to go to this picture, and this is G2 of interphase, the first one, and that's just showing you this is preparing to divide. In prophase, a couple of different things happen that are really important. One, the DNA condenses into those really thick chromosomes that you see there. Okay, the spindle fibers start to form. You can see those in yellow, and they're attached in animal cells to centrioles at the ends. And then the other thing that happens is a nuclear membrane begins to break down. Feel free on your note sheet to fill that in under prophase. That the nuclear membrane breaks down, the spindle fibers begin to form, and the DNA condenses into those chromosomes. In prometaphase, we're still going to be breaking down that cell membrane, or sorry, not cell membrane, the nuclear membrane, releasing those chromosomes out of there, and they're going to attach to the spindle fibers. and they're going to migrate towards the center of the cell. Why? Because in metaphase, they're going to line up right along the middle of the cell. They're going to be attached to the spindle fibers in one, and then the centrioles at the other. And the centrioles are called centrosomes when they're attached to the spindle fibers. I think is the difference between centrioles and centrosomes. Anaphase, they're going to start pulling those chromosomes, those sister chromatids apart. Remember, one half is a copy of the other. So when you pull those sister chromatids apart, you're just taking one copy and going to one side of the cell, and the other copy and going to the other side of the cell. And that's just going to keep happening. That it's going to keep elongating. Notice the cell's a little more oval now and a little less round. And it's going to keep stretching and stretching and stretching until at some point we go into telophase, where the chromosomes on one end of the cell are going to gather around and we're going to reform that nucleus. And the other end is going to do the same thing. And we're going to dissolve the spindle fibers and we're going to pinch off that cell. Cytokinesis is actually defined as the division of the cytoplasm. So cytokinesis isn't really a stage, it's a process that's happening during telophase. And this just shows it to you in plant cells. We will look at some of these underneath the microscope. These are onion roots, because the root of the onion is growing continuously, so they've got lots of cells there that are dividing. You can see here the first picture is prophase. You can see the stained dark blue. Those are the chromosomes inside the nucleus. And you can see the nucleus is kind of stretched out in the cell. It's breaking down. Prometaphase, it's breaking down even more there. Metaphase, the chromosomes are in the middle, and anaphase are starting to pull away from each other. And then in telophase, instead of pinching off the cytoplasm, a cell plate forms, and that will eventually become the new cell wall. And this is just a kind of some really good images that are I can't think of the word right now. That are they're color coded, but they're not stained. These images are from probably like some other. I'm just not like bringing the words today. These are probably like an MRI image or something like that. And you can see those spindle fibers there in green and the nucleus in dark blue. You can see in the prometaphase, you can see where they look more like chromosomes instead of just a mass of DNA in the center. I think there's more of those. Oh, come on. Okay, there we go. So metaphase, anaphase, and telophase. You can see them splitting apart, and then the last one kind of becoming two cells there. This just goes over some of the kind of the vocabulary. You can see the sister chromatids, the spindle fibers attaching to it. These are a metaphase plate there where they're kind of congregating there in the middle. The kinetochores are the half that is on one sister chromatid that is pulling in one direction. The centromere is the center of those two sister chromatids, and it connects those kinetochores. And this is showing that again. They did an experiment with this, and this is how they know how this actually happens. You can see there's like the microtubular, the spindle fiber actually revolves around a motor protein that actually literally like tugs and pulls, kind of like a pulley system, pulling those chromosomes from one side or the other. So let's see how far we got. Okay, there we go. So cytokinesis in animals, a cleavage occurs beginning with a cleavage furrow, and in plant cells they produce a cell plate where the cell wall will eventually form. You can see the difference here between the cell plate and the cleavage furrow. Binary fission is an asexual reproduction of the single cell. That should say single cell. Oh yeah, single cell eukaryotes. So sometimes you're dealing with single cell eukaryotes and not just bacteria. So binary fission can also refer to bacteria, but it can also refer to like an amoeba splitting and making a whole new organism. Does not involve mitosis. Typically, bacterial DNA is a single circular piece, so this step is not necessary, as well as amoebas. They just don't have enough DNA, and so they don't go through the whole chromosome splitting process. And this just shows a picture of bacteria doing some binary fission. The evolution of mitosis. Eukaryotes precede prokaryotes. That means that eukaryotes came after prokaryotes. We might hypothesize then that mitosis evolved from simpler prokaryotic mechanisms. Some of the proteins involved in binary fission are related to eukaryotic proteins their function in mitosis, which supports that hypothesis. Eukaryotic cell regulation. The cell cycle is regulated at certain checkpoints by both internal and external signals. The checkpoints are control points where stop and go ahead signals can regulate the cycle based on whether crucial processes have been completed correctly. The three major checkpoints occur in G1, G2, and M. If the cell does not receive the go-ahead signal at this point, it will exit the cycle, switching into the G0. Most cells in the human body are actually in a G0 state. Many of our cells don't divide ever or infrequently. So these are those checkpoints, and they're basically like a regulation. They check to make sure the cell has done what it's supposed to do. If it has not, it's out, it's done. It could die. It could go through what we call programmed cell death or apoptosis. If it's good and everything has happened appropriately, then it will go on and go through that division process. Cyclins and cyclin-dependent kinases. The pace of the events of the cell, these proteins pace the events of the cell cycle. The protein kinases, remember ACEs, that's going to be an enzyme, are enzymes that activate or inactivate other proteins by phosphorylating them. Think about our cell communication stuff that we did with chapter 11. Okay, remember that phosphorylation cascade where we're taking ATP and taking that phosphate off and donating it to the process. Some protein kinases give the go-ahead signals at the G1 and the G2 checkpoints. Some kinases must be attached to a cyclin to be active. That's why we call them cyclin-dependent kinases, or CDKs. MPF is a CDK that was discovered first, and we saw that in frog eggs because frog eggs are transparent. So they were, well, I say we, not me, scientists were able to see that because frog eggs are transparent, and so they could see what was going on in those, and they discovered the MPF cyclin-dependent kinase first. And this just shows you a fluctuation of the MPF activity and cyclin concentration during the cell cycle. You'll notice that when you're at those that M phase, you'll notice that those enzymes are going to go up. Molecular mechanisms that help regulate the cell cycle, and so this just shows you again what's happening here. Those CDKs are checking for making sure that everything is occurring correctly, and if it is, the cell can proceed. If it doesn't, the cell cannot proceed. When cyclins accumulate, phosphorylation occurs, and that's going to initiate mitosis. So how does it go about saying, okay, you're good? Well, when those cyclins accumulate, and there's lots of them, then we're going to have lots of phosphorylation, and that's going to actually be like a trigger, a cell communication that says, here's the response you need to have. And the response is to go ahead and go through that division process. M checkpoint is an internal signal example. Anaphase does not begin until all the chromosomes are attached to the spindle. If they are, then a series of molecular events occurs to proceed. Growth factors are an external signal example. Different cell types respond differently to specific growth factors. That would maybe trigger them to either, you need to divide, or, oh, you're a cell that doesn't need to divide. They're not going to get those growth factors. Density-dependent inhibition is an external signal where crowded cells will stop dividing. And this gives you an example with, I think, a human sample of connective tissue is cut up into small pieces. Enzymes digest the extracellular matrix, resulting in a suspension of free fibroblasts. Cells are transferred to culture vesicles. PDFG is added to half the vesicles, and then they can watch that division process. And then this one here is anchorage dependence, density independent inhibition. So they get really, really thick and they will stop dividing. But if you take some out, they'll divide more to kind of fill that space. Cancerous cells are a problem. You can see over on this right-hand side because they continue to divide even though there's not really enough space. They're going to continue to divide anyway. We're going to talk about cancer here. So here we are. Cancer is a loss of cell control. Cancer cells do not accept the normal signals that regulate the cell cycle. Cancer cells divide excessively and invade other tissues. Invading other tissues is kind of important there. Underlying issues is that nearly always a change has occurred to one or more genes, and that alters the function of their protein products, resulting in loss of control over the cell cycle. When cancer cells do not stop dividing, they do so at random points. Some never stop dividing. HeLa cells are a really good example of this. These are there's a strain of cells from, and I think we'll read a little bit about this in an article, but they're called HeLa because they came from Henrietta Lacks in the 1950s at Johns Hopkins. There's a great book on this. If you've never read The Immortal Life of Henrietta Lacks, it's an excellent book. and it talks about the historical aspect of what was going on with Black Americans in 1950s at Johns Hopkins, as well as the cellular science of what's going on. But she had, I'm trying to remember right now, cervical cancer, and her cells were taken without her knowledge. And they have grown and grown and grown. You can actually purchase strains of HeLa cells, but they've over time they've mutated. Also, there's multiple strains of these, but these cells, even though she's long gone, they are continuing to survive and live and divide because they don't respond to normal signals to stop growing. Cancer cells also do not undergo apoptosis or programmed cell death when something goes wrong. So normal cells, if something goes wrong in the cell, it kills itself. It's what we call programmed cell death. It's designed to die. But cancer cells, oftentimes there's something wrong with them, but they don't die when they're supposed to die, and so they over produce and they create tumors. Typically, if a cell transforms into a cell that could create a problem, the immune system destroys it. But in the event that it does not, the cells may divide uncontrollably into a tumor. A benign tumor is non-cancerous. That means those cells are kind of isolated to themselves. They're not invading other tissues. And the malignant tumors are ones that those cells are actually problematic because they could invade other tissues and create more tumors somewhere else. Metastasis is when cancer cells spread from one location to a more distant location. Once a cancer has what we call metastasized, it is much harder to treat because it's like chasing a moving train. It just keeps going and you can't catch it. So this is showing a tumor in breast cancer. A tumor grows from a single cancer cell. Cancer cells invade neighboring tissues, then they spread through the lymph system or the blood vessels to other parts of the body, and now the cancer may survive in another part of the body. So it's like chasing a moving train. You get rid of the breast cancer, but now you pop up with a tumor 6 months later in your liver, and you get rid of that, and those cells, by the time you discover the tumor, are already somewhere else in the body, and they're going to be creating a tumor there. So you're just chasing this thing all over the body. Metastasizing is not a good thing. When that happens in a cancer patient, that means that it's going to be much, much harder to treat. That's usually when they introduce chemo. So why the cell cycle? Why do we care about the cell cycle? There is so much to learn about cancer to treat it, but in order to do that, scientists must understand better the role of the normal cell cycle. If we don't understand how a normal cell responds, then there's no way to understand how an abnormal cell responds. And I believe that is the end of our presentation, which is probably good because it was 22 minutes.